Brief moment to reflect and celebrate the incredible talent among the faculty of WashU Medicine, and that is why we are so grateful to our donors who make these professorships possible. Today, we have the privilege to recognize the exceptional talent and accomplishments of Dr. Yoel Ju. Because we marvel at your career, Yoel, this is such an honor for us to be able to celebrate what you have done. And I also want us to recognize your family. Yoel is joined today by her children, Nora and Owen, and her sister, Seal. And I know you have many friends and colleagues joining you today as well. Could I ask the four of you to stand up so that we can all celebrate and applaud? We are live streaming the event, and for those who are unable to join us in person, it's my understanding that several of your other friends and family members are joining us virtually. And before we do the formal academic introduction and bestow the medallion, let me share a few highlights about our donor, in this case, Barbara Burton and Reuben M. Morris, and their legacy at Washington University. Philanthropy and generosity were hallmarks of the lives of Mr. and Mrs. Morris, whose investment in Washington University, and specifically in Alzheimer's disease research, continue to make a profound difference today. Their legacy at the university began when Reuben graduated from the Washington University School of Law in 1964. Following his graduation from the law school, he joined Boatman's Bank and remained at Boatman's Bank, becoming president until his retirement in 1995. Throughout his career, Mr. Morris was an active local leader and philanthropist. He was chairman of the board of Mary I and St. Louis Country Day School and William Woods University in Fulton, Missouri. He served on the board of St. Luke's Hospital and the St. Louis Bi-State American Red Cross, and was treasurer of the Episcopal Diocese of Missouri. Barbara Burton Morris was active as well, serving on the boards of the Alzheimer's Association and the Central Institute for Death. In addition to their many St. Louis philanthropic commitments, the couple maintained strong ties to Washington University. Mr. Morris served on the university's Alumni Board of Governors, the law school's National Council, the University Campaign Cabinet, and the Law Alumni Association Board. In 2000, he received the Distinguished Law Alumni Award, and throughout their lifetime, Mr. and Mrs. Morris supported Washington University School of Law, Siteman Cancer Center, and what is now called the Joanne Knight Alzheimer's Disease Research Center. In 2018, upon Mrs. Morris's death, the couple left a generous bequest to the School of Medicine with the purpose of advancing Alzheimer's disease research. A portion of this gift established 3 endowed professorships, each of which bears the name and memory of Barbara and Reuben Morris. These endowed chairs are truly testaments of the couple's deep commitment to the School of Medicine and the pioneering efforts that are going on in our Department of Neurology and in neurosciences in general that are so important to where we are with respect to improving human health. So let's have a round of applause for the Morris family. And now we get to proudly install Dr. Yoel Ju into the second of the 3, Barbara Burton and Reuben M. Morris, 3 professorships. In a moment, I will ask Dr. Jinmu Lee, chair of the Department of Neurology, to give the more formal academic introduction. But I just want to say I had the privilege of hearing about Yoel's research on numerous occasions as a part of our personalized medicine initiative. And her research on disordered sleep and how it impacts Alzheimer's disease And every time I hear more about what she's doing, I am just so excited about the potential. I've been so impressed by how her work brings together rigorous clinical research into a fascinating area and something that has such an enormous role in health and disease, how we sleep. Yoel, we're so excited about the work you are doing and so happy to be here today to celebrate it with you. And now I want to ask Dr. Jinmu Lee, the Andrew B. and Gretchen P. Jones Professor of Neurology and Chair of the Department of Neurology, to give the formal academic introduction.
So it's an absolute pleasure for me to introduce Dr. Yoel Ju, who is being installed today as the Barbara Burton and Reuben M. Morris Endowed Professor. So Dr. Ju comes to us at Washington University after quite an international journey. She was born in South Korea, and this makes me very proud because I'm a fellow countryman from South Korea, and spent some time in her early years in the Republic of Kenya. Then she came to the United States for high school, and then she matriculated at Harvard University, where she majored in biology, graduating magna cum laude. Afterwards, she went to Columbia Medical School, where she was inducted into the AOA Honor Society, and we're so lucky that she chose to come here for her neurology residency, and we never let her go. So we're so glad that you're still here. She also completed her fellowship in sleep, as well as a fellowship year in research, where she completed a master's in clinical investigation, and she joined the faculty in 2011, rapidly rising the ranks to full professor in 2020. So Dr. Ju is internationally recognized for her studies relating sleep to the pathogenesis of several neurodegenerative diseases, including Alzheimer's disease. She's published several landmark studies supporting a link between sleep disturbance and AD, Alzheimer's disease pathogenesis. In 2013, she was a part of a team that published a paper in JAMA Neurology demonstrating an association between sleep quality and amyloid deposition in the brains of volunteers with preclinical Alzheimer's disease. That's before they develop symptoms of dementia. Amyloid, of course, is one of the pathological hallmarks of the disease, and so a biomarker, a marker of the disease. Then in 2017, she published another paper where she demonstrated that disruption of slow-wave sleep resulted in alterations in A-beta. A-beta is the peptide that aggregates to form amyloid plaques. And its presence detected in the CSF in normal individuals. Then in 2017, she demonstrated this link between slow-wave sleep and Alzheimer's disease. And this was incredibly important because it identified a specific phase of sleep that was most closely associated with the A-beta levels in the CSF. And we know that there are drugs that can enhance slow-wave sleep, so it opened the door for future interventions. Finally, she published a series of papers demonstrating an interesting but complex relationship between sleep apnea, a sleep disorder that disrupts slow-wave sleep, and biomarkers of Alzheimer's disease. And that treatment of sleep apnea resulted in decreases in CSF A-beta levels. Again, very important, linking slow-wave sleep specifically to Alzheimer's. Dr. Ju has since expanded her scientific pursuits beyond Alzheimer's disease, also studies sleep and its relationship to other neurodegenerative diseases, including Parkinson's and other synucleinopathies. She's been extremely successful with grant funding, currently holding 2 R01s and a U grant. She's PI of a study called NAPS, which is a prodromal study of subjects with synucleinopathy. She's won numerous awards, including the Glasgow-Reuben Achievement Award from Columbia, the World Association of Sleep Medicine Young Investigator Award, and the Wayne Henning Sleep Investigator Award from the American Academy of Neurology. She's clearly an internationally recognized expert for her groundbreaking work on the importance of sleep in Alzheimer's disease and other neurodegenerative diseases, and her work has convinced me how important sleep is. So I try my best. So Yoel, it's an honor to have you on our faculty, and congratulations on your installation.
This is only gold. I mean, those and it reads, Barbara Burton and Reuben and Morris, third professor, Washington University. And on the back it says Yoel Ju, MD, September 19th, 2023. We are so proud of you, Yoel.
All right, well, thank you so much for that very kind introduction, Dr. Lee and Dean Perlmutter. And I wanted to thank the Morris family for the generous bequest towards Alzheimer's disease. I am very grateful and honored to be continuing their legacy. I'm going to be telling you about my work in sleep and neurodegeneration, which encompasses Alzheimer's disease, but as Dr. Lee mentioned, other neurodegenerative diseases as well. So I'm going to start with a background, and I will preface this by saying that I know there are a lot of people here as well as watching online who are not scientists, and so I've tried to keep this as interesting for them as possible. So we're not going to go like deep into the meat of the data. This is going to be all fun stuff. So I'm going to tell you a little bit of background about me. So that's me and my sister when I was little in Korea. I'm the one with the pigtails. I love those overalls or whatever I'm wearing. Those are awesome. And then this is my passport picture from when I was 5. And when I was 5, I moved to Kenya. And this is us. We stopped at the side of the highway to take a picture at the equator. This is the best picture of us at the equator. And I'm like, this is so bad. And then I had 2 children, and I realized you can never get both children looking in the same direction. So this is the best we've got. This is us riding a camel. This was a one-time occasion. We did not do that regularly. And I went to boarding school from 3rd grade. This is me there. And I put this to remind myself that in 3rd grade, I got my only grade of a C, and it was in science. But it was an interim report card only, not a final one. But I think that back then, with a C in science, I don't think I could have foreseen that this is where I would end up. And then I moved to Los Angeles for junior and senior years of high school. And when I was a senior, I was a part of the academic decathlon, like a good dork. And the special topic for that year was neuroscience or brain science. And that really sparked an interest and love of neuroscience that has persisted. And that's my mom in that picture. And then I went to Harvard for college, and I was going through all of my photos, and I realized I have surprisingly few photos that are appropriate for showing at an installation. And like, thank goodness there were no iPhones or Facebook when I went to college. It's bad. I worked in a basic lab where I did research on deformable substrates for in vitro neuronal growth with Lisa Flanagan, who's now at UC Irvine, and this is me getting my neurobiology certificate. And I graduated. And at that point, I kind of wobbled on whether I would get an MD PhD or just an MD. And I'm kind of ashamed to admit I did just the MD and have never made up for the PhD. But I do have lots of friends with PhDs, like my sister and my friend here, Liz. So I have a lot of PhD friends, so I feel like that makes up for it. Then I went to New York to Columbia PNS for med school, and I think this was like the transition from regular photos to digital photos, because I couldn't find any except these. This is on the roof of the med school dorm. This is actually where I watched the Twin Towers fall, like 3 weeks after I started med school. So I think I'll always carry a little bit of New York City in my heart. This is me with my friends. I'm dressed up as Smurfette. I made that outfit, including the hat. And then I would say the highlight of my time in New York was going to Christie's, the auction house. They had a show where they had Phineas Gage's skull on display. So Phineas Gage was a very famous case in behavioral neurology. So apparently it was enough for me to take a picture that day. Throughout med school, my interest in neurosciences and neurology kind of sharpened, and I came to WashU for residency. This is a picture of my ID. I have to walk around with this ID with a picture of my 26-year-old face next to my current 44-year-old face. So all that optimism, hope, energy. This is me on the last day of residency. I was on a month of every other night call as chief resident. This is what happens, and this is kind of like emblematic of residency. I actually put that up to show how long I've been here. That building was knocked down, and then there was a giant pit in the ground for a couple years where an entire ecosystem formed with frogs and birds and whatnot. And then one day they turned it into the Whole Foods on West Pine and Euclid. So I've been here since it was the medicine shop. And residency was very demanding, but we did have some fun. So this was the residents who put together a dinner for Dr. Sohn when he retired. And I'll say Dr. Sohn wrote a letter for me to join faculty, so thank you, Dr. Sohn. And there's still quite a few people here: Arun Vardachari, Bob Buccelli, Gabby DeBruin, Alex Carter, Rachel Darkin also. And then me, and then this is Brian Somerville, who was my classmate at med school and co-resident in neurology and colleague in neurology and a spouse for many years. And now we are not those things, but we are still co-parents to our wonderful children. And this is us going to one of the 3 World Series games, or World Series that the Cardinals have gone to since I've lived here. And then in my free time, I really like to knit. So this is part of my knitting group on a yarn crawl. And I won some ribbons at the Missouri State Fair. And I think we should get this sign that says Wall of Winners and put it up in our department with our pictures. All right, so that was at residency. And then I started fellowship here in sleep medicine. And I did a year of clinical sleep medicine where I cared for patients with sleep disorders, including people who had neurodegenerative diseases or dementia and had a lot of sleep problems related to that. In my research year, I focused on a disorder called REM sleep behavior disorder. And this is a disorder where people act out their dreams. And the dreams are typically where they're defending themselves against an attacker or an animal. And while that's interesting in itself, it's extra interesting because there is a strong link with neurodegenerative diseases. And so during fellowship, I published a large case series about changes in the demographic profile of RBD. Here's me presenting a poster. Here's Steve Duntley and Linda Larson-Pryor, who were my mentors for that RBD project. And I became interested in multi-center RBD research. So I'm gonna talk a little bit about RBD, and just for interest's sake, I'll tell you about a case. And in fact, I still see this patient 10 plus years later. So at the time, she was 70, and she had had 7 years of recurrent nightmares of being attacked, but there was no inciting event, they just started. She has associated vulgar screaming and movements. She usually sleeps alone, but she is with her dog, but she has awakened to find she was strangling her dog. And she came to clinical attention because she went on vacation, shared a room with her granddaughter, who the next morning said, "Grandma, you were saying bad words in your sleep." She has no other sleep disorders. Social history is really, very high functioning, well-educated woman, and she had a normal physical and neurological exam. And when you come to the sleep center, you get a sleep study. I'm gonna try to play this. So the things that we look at during a sleep study, the blue lines are the eye movements, and you can see these sharp sort of pointy things that tell us that the eyes are moving rapidly. It's a rapid eye movement sleep. This is the muscle tone and the chin. These are the brain waves, the electroencephalogram or EEG. These are leg movements. This is microphone or for snoring, airflow, the breathing effort, oxygen, and heart rate. So we monitor a lot of things. And every 30 seconds, the technologist who's reading this assigns a stage. So this says R for REM sleep. Is the sound working? Do people hear anything? So when you're sleeping, you're supposed to just lie there immobile, right? She has this thing on her finger that's bright. It's measuring her oxygen. You can see she's moving around, and she's starting to talk. Can the AV person turn up the volume?
My white sofa, my good sofa, it's ruined.
All right, so she's talking about her white sofa, and then she's quiet for a while. I think the person in her dream is responding to her. And you can see she still has these rapid eye movements. She's clearly in REM sleep. So right now, she looks like she's asleep, but then she'll start up again.
Here's the other one. I'll tell you what to do. I swear to God, if you go doing that one, I'll kill you. Why did you pile pillows here? The pillows are full of shit.
Sorry, I should have told my kids to cover their ears. So this is not how she behaves when she's awake. She's a lovely, very gentle person when she's awake. So she kind of has this very different personality and fighting back against someone who is wronging her on her couch. So this person has REM sleep behavior disorder, where people act out their dreams. So this wasn't one of her typical dreams that she came with, but that's actually quite common for people to have multiple types of dreams that they come in with, but the ones that they come for are the scary ones. And RBD is usually noted or diagnosed in people who already have a known neurodegenerative disease. And in studies where they have looked at the brains of people with RBD and neurodegenerative diseases, 98% of them have something called a synucleinopathy. And this includes 3 different diseases that are very different. So Parkinson's disease, which is a disorder of movement, dementia with Lewy bodies, which is a disorder of thinking, and multiple system atrophy, which is a disease of the autonomic nervous system. But all of them share a common appearance under the microscope, where they have these clumps of a protein called synuclein. And so altogether, they're called synucleinopathies. Some people have RBD on its own. We call it idiopathic. But when you follow these individuals, 6 to 8% per year are diagnosed with a synucleinopathy. So here's a large multi-center study that looked at the proportion of people who don't have one of those disorders, and you can see that over time, almost all will be diagnosed with a synucleinopathy. And so we think of people who have just RBD or idiopathic RBD as having a preclinical or prodromal synucleinopathy, meaning they already have the disease in the brain, but they don't have overt symptoms of the synucleinopathy yet. And so this is a window of opportunity for potential intervention, because if you could stop the synucleinopathy process in the brain, potentially you could treat them so that they don't get it and have just the RBD, which itself can be treated. So I was still a fellow, and I kind of had swirling and marinating in my mind this thinking about preclinical neurodegenerative diseases and how sleep relates to that, when for a research conference that we had, Dr. David Holtzman gave a talk. Unfortunately, Dr. Holtzman couldn't be here today. He is traveling to give his own lecture, but I wanted to acknowledge the amazing mentor that Dr. Holtzman has been, as well as the incredible scientist and example he is for me and many of us here in the room. So Dr. Holtzman presented this paper where, from his lab that is very famous now, I think all of us in this room have seen this, where as mice, so all the data about mice have a mouse and all the humans ones will have a human. As they are more awake when the light is dark, or when it's dark, this level of a protein called amyloid beta goes up. And as the mice sleep, the level goes down. And so this up and down that correlates with the waking was very interesting. And so I wanted to back up for the people who are not neurologists to describe what neurodegenerative diseases are and how we think about them. So these are diseases where previously healthy neurons die. The risk of them increases with age. About 7 million Americans currently have a neurodegenerative disease, and it costs us $500 billion a year currently. And something that is very common among very different diseases is that proteins accumulate or aggregate, clump together abnormally. And so because of that, we often group neurodegenerative diseases by which protein clumps up. So I've put here, so synuclein is a Parkinson's disease and dementia with Lewy bodies and MSA. And Alzheimer's disease is half of the neurodegenerative diseases. And in that disease, amyloid clumps up, and tau is the other protein. There's two proteins involved. Now, Alzheimer's disease, unlike RBD, is exceedingly common. We are expected to, we currently have about 6 million people with Alzheimer's disease in the United States. And by 2050, it'll be close to 14 million. This will bankrupt our healthcare system unless we have better methods of detecting and treating Alzheimer's disease, hopefully before its symptoms start. And so Alzheimer's disease is a much more, I guess, important problem from a public health standpoint. And also, at the time that I saw the data that Dr. Holtzman presented, I was at Washington University, which is an incredible hub of Alzheimer's disease research, and I thought, maybe I can learn something from these Alzheimer's disease researchers. Something that the people at the Alzheimer's Disease Research Center here and others have described is that there is a very long preclinical phase of Alzheimer's disease where, so down here is the symptoms, so in the middle is when symptoms start. There is probably 10, 15, 20 years where amyloid plaques are forming and tau tangles are also forming in the brain, but people have no symptoms. However, there are biomarkers that have been identified and developed in Alzheimer's disease that can tell us if someone has Alzheimer's disease in the brain and how far along that is. And the important thing about the preclinical phase is that it's a window for treatment where if we can identify them and treat them appropriately, that they won't get dementia, which would be great. So with that background, I'll show you the data from Dr. Holtzman's lab. So again, the amyloid beta increases during wake and decreases during sleep. They then gave the mice orexin, which keeps people awake, and they found that the amyloid levels did not go down as predicted. When the mice were chronically sleep deprived, they got these black splotches, which are amyloid plaques, compared to the controls, which really didn't have as many. So in my naivete, I said, well, what about in people? And he said, well, no one knows. Do you want to find out? So he very, very smoothly convinced me somehow to do this study with at the Alzheimer's Disease Research Center, looking at whether there's a relationship between sleep and amyloid. And so what we did is we had around 200 people wear an actigraph, which is a motion sensor that can tell us about people's sleep quality and quantity. And then we compared that with who had amyloid plaques as determined by a test in the spinal fluid. These are all participants at the research center who are followed for years, and so they'd all had this testing done. And we found that the really good sleepers compared to the bad sleepers down here, the bad sleepers had 5.6 times risk, after accounting for other variables, of having amyloid plaques. These were all completely normal people. They had been tested very extensively. These are normal people. However, we know that the people with poor sleep were much more likely to have amyloid plaques, i.e. Alzheimer's disease, starting in the brain. A few years later, with Eric Music, we looked at the rest activity rhythm over the 24-hour day, and we found that the more fragmented, meaning more switching between rest and activity someone had, that's on here, the more Alzheimer type biomarker levels they had in their spinal fluid. So then we were kind of faced with this, well, there's this relationship between sleep disturbance and Alzheimer's disease, and like, what's that about? And so then, since then, I think in the past 10 years or 13 years now, we've spent trying to translate the findings from that original mouse paper into humans. So one first thing that came up very soon from the lab of Randy Bateman is that humans also have this cycling of the amyloid beta levels. That's the double line here. There's two types of amyloid beta. However, in this study, the humans had a lumbar catheter in the lower spine to sample their spinal fluid. And so there's a little bit of a lag because the proteins that you're measuring need to travel from the brain down to the lower back. And then sleep deprivation increased amyloid beta, and a few years later, Brendan Lucy published this finding from this really, truly heroic study where people had lumbar catheters, and some were sleep deprived, some slept normally, some had a sleep promoting drug, and the folks in red, they have an elevation in the amyloid beta. A similar study was done in the Netherlands. The first author is Sharon Ohms, who actually ended up being a visiting student in my lab, and they also found a difference between the sleep-deprived people. They had a higher level of amyloid beta. So we know that sleep deprivation increases amyloid beta. But the question is, well, which part of sleep? So sleep is quite complex. There's non-REM sleep, there's REM sleep, there's different aspects of sleep. And one aspect of sleep is slow-wave sleep, and that's the deepest non-REM sleep you can get. Here. So this is the brain waves, the EEG, these nice big, like these things make me relax just to look at them. So a nice deep sleep. And what we found is that in normal individuals, the more of the slow-wave sleep you have, cross-sectionally, the lower your amyloid beta levels. So this gave a hint as to, oh, well, maybe it's a slow-wave sleep. However, we wanted to confirm that. And so we disrupted slow-wave sleep to see if that increased amyloid beta. So what we did is we enrolled, I believe, 22 people. We had them stay overnight. Two times a month apart, and it was randomized which condition they had. One of them, they had a slow-wave sleep disruption where we would read the EEG, we would transform the data, look to see if it was artifact, if it was real data, and then if they had slow-wave activity, we would give them a little beep through headphones. Every 10 seconds, this beeps would get louder and louder and louder to the maximum permitted by the Human Research Protection Office. On average, each person got 1,200 tones over the course of the night. It is very difficult to get people out of slow-wave sleep. Their brains want to sleep, so it's very hard to wake them up without completely waking them up. And then we compared it to another night where the mute button was on. And what we found is that the more we were able to disrupt their slow-wave sleep on the x-axis here, the more their A beta increased compared to the sham night. On the other hand, their total sleep time, the amount of time in non-REM or REM, did not have a relationship with the amyloid beta levels. And so slow-wave sleep really seems to be the specific thing that is linked with amyloid beta. We also compared it with other proteins, and really its A beta is very specifically correlated with slow-wave sleep. So while I was actually writing the scripts for the slow-wave sleep disruption, I made another slow-wave sleep disruptor. This is Nora. So my student, Sharon Ohms, at the time would actually ferry a laptop back and forth to my house while I was on maternity leave so I could tune the parameters on the computer so that we could slow wave disrupt. Nora has spent a lot of time in my office. She's a month old here. I also think of her as my K-23 baby because I had been on an institutional K award, which is a career training award, and then I wrote my K-23 and submitted it when I came back. And I thought all the neurologists would appreciate the shirt. It says, because I'm the neurologist, that's why. I also wanted to mention tau. When we looked at the actigraphy, meaning the activity levels at home that can tell us how well people are sleeping, we just compared the week preceding each of the months, the nights that they came in a month apart. And we found that the worse that they slept at home on one night compared to 1 week compared to the other week, the higher their tau was. So there seems to be a relationship between tau and sleep quality as well. Several years later, again, Brendan published this finding in humans where the sleep-deprived individuals had an acute increase in tau, and the same was shown in mice as well, where the red are the sleep-deprived mice, and they have higher levels of tau. So when we think about the relationship between sleep disturbance and Alzheimer's disease, there's probably a two-way relationship. And based on the data we have so far, we know that sleep disturbance causes decreased slow wave activity, which leads to increased amyloid beta release. Amyloid beta is a soluble form of amyloid. And there's probably decreased glymphatic clearance as well. This is the waste clearing mechanism in the brain. And together, we think this would increase amyloid beta levels, which over years would increase risk of amyloid plaque formation. Also, sleep disturbance, we know, increases tau, which interacts with amyloid plaques and eventually leads to neurodegeneration in Alzheimer's disease. I think I'm going to go really fast on the obstructive sleep apnea part just because of time. I then looked at individuals with obstructive sleep apnea, which is a common sleep disorder. That's not a brain disorder. People just stop breathing in their sleep because of an obstruction in the airway. And what we found is that these individuals seem to have difficulty clearing waste products from their brain space into the spinal fluid. And they also had an abnormal relationship between their slow wave sleep and their amyloid beta levels. But once we treated them, we were able to rescue them, i.e. It restores the relationship between slow wave sleep and amyloid beta. To this cascade, obstructive sleep apnea seems to contribute in multiple ways to increase the risk of Alzheimer's disease. Obstructive sleep apnea is linked with a higher risk of dementias down the line. We know that there is this reverse direction where people who have changes in their brain of Alzheimer's disease have injury to sleep and circadian regions that then promote sleep disturbance. Amyloid plaques actually directly decrease slow wave activity. And then as people develop even subtle symptoms of Alzheimer's disease, they have changes in their activities, and also people tend to have more medications that also contribute to sleep disturbance. So there's multiple feedback loops in the relationship between sleep disturbance and Alzheimer's disease. That tends to perpetuate and make it worse and worse. So once people actually have dementia, many of those individuals have sleep problems. And actually sleep problems in dementia are the primary reason that people get institutionalized in nursing homes in dementia. I think the optimistic viewpoint, though, is that each of these arrows represents a place that we can intervene. So that we can break some of these feedback loops, hopefully improve people's sleep, which feels great, but also decrease the risk of Alzheimer's disease. This is, a lot of this work went on in my early attending life. And then I had a second slow wave sleep disruptor, and this one I call my R34, R01, RF1 baby, because I had still been involved in REM sleep behavior disorder research and had put together an R34, which is a very small multi-center grant to help plan clinical trials, and that funded 2 months before my due date. And so there was a lot of planning we had to do to roll out the study. I also submitted my R01 the day before I left for maternity leave. There's no deadline like a scheduled C-section. And then I wrote an RF1 together with Dave Holtzman and Eric Music, which was submitted shortly after my return. If anyone needs to remember when the last total solar eclipse was, it was in 2017. And he gets to wear this shirt too, and he also gets a hand-knitted going home outfit. So that's Owen. So then I wanted to shift gears and talk about my work in REM sleep behavior disorder and the NAPS consortium. NAPS stands for North American Prodromal Synucleinopathy Consortium. I did not come up with that acronym. Ron Pastuma did at McGill. It is brilliant. The NAPS consortium, which was initially funded with that small R34 grant, enrolled people with RBD at 10 different sites across the US and at McGill. And the main thing that we did during that grant was to come up with a standardized way to assess these patients across multiple sites. Because Parkinson's disease is usually evaluated by movement disorder specialists, dementia is usually evaluated by dementia specialists, and so on. Each center was kind of doing their own thing, doing great work, but doing slightly different things. And so what we did in NAPS is said, okay, this is how we're all gonna do it. The motivation for doing NAPS was so that we could one day see a patient who has RBD and hopefully figure out which pathway they were going to go down. So these are potential changes in clinical markers and biomarkers in Parkinson's disease, dementia with Lewy bodies, and multiple system atrophy. And RBD is present in each of these. And we think that in each of these disorders, again, there's that window of opportunity in the preclinical or prodromal phase where we can intervene and prevent people from getting the overt neurodegenerative disease. In NAPS1, we enrolled over 360 participants, and this is data from the first baseline paper that described the cohort. And we did a variety of neurological tests on them, and what was really striking is that in this normal group of people, you talk to them, they are usually very well educated, like they seem normal. However, when you really test them, we find broad abnormalities in thinking. So this is a thinking test. All the black bars are abnormal scores. So all these people have abnormal general cognition. This is a MDS-UPDRS is a test of movement, and about a quarter of individuals had abnormal total scores on that. And then this is the FM100, which is, I put a picture here. It's a color vision score, and you have to put all these little circles in order of the color. And about half of the people in our cohort couldn't do this because their color vision is impaired. And so we find these broad, subtle neurological deficits, and what we really want to know is, well, how does this change over time, and how can we use this type of information to predict when they're going to develop a synucleinopathy, which one, or for the lucky few, if they aren't going to at all. And so then we wanted to expand the study into a longitudinal study to follow them up. And then we had another slow wave sleep disruptor, which I describe as COVID times. These are seriously the only two pictures I took between March and June. This is my daughter reading Harry Potter, and I don't know what Owen is doing here. It does not seem safe. But COVID was a hard time for parents of young children. So he was 2.5 and she was 5. So those were very busy times. I also wrote a U19 for NAPS, and that was submitted in June of that year. So I spent many hours working on that. And then after it was submitted, then we could have some happy time. So that's July 4th, obviously. The goal of NAPS continues to be to prepare for clinical trials. We maintain a trial-ready cohort of at least 300 people with RBD and a NAPS RBD registry. And we are very interested in developing biomarkers for use for clinical trial participant selection, i.e. who's gonna benefit most from a certain drug once we come to clinical trials, but also developing markers and measures to use as outcomes. So because we don't want a clinical trial where we're waiting for people to develop Parkinson's or dementia. We want to be able to do short trials where we can tell if a treatment is effective quickly. And we are collecting all of the data and samples so that they're shareable publicly with other researchers and groups and individuals. So we applied for NAPS2. NAPS2 is much larger. All of the sites are here. But to accommodate the much larger protocol, we have 8 cores that process the different types of data that we're collecting. And these all feed into a large project, which is predicting phenoconversion, meaning how can we use the data we collect to be able to predict in an individual what their outcome is going to be. We also have a recruitment, education, and outreach core that is outward facing, that interacts with foundations, industry partners, and so on. NAPS2 is approximately $35 million. It is co-funded by both the NIA and NINDS at the National Institutes of Health. The protocol is really expanded in NAPS2. We're seeing people back longitudinally, meaning annually, for the extensive neurological testing. We've also added brain scans, MRIs, and DAT scans, which will get dopamine activity. We're doing sleep studies called polysomnograms, and we're collecting blood on everyone, and in those who are willing, we collect spinal fluid with a lumbar puncture. And then we also will be enrolling control participants who don't have RBD. One of the things that we're actively working on right now is a clinical scale to rate people in all of these different neurologic domains so that we can capture the very gradual and subtle change in people's clinical status across these domains prior to them phenoconverting, meaning developing a synucleinopathy. And this was directly inspired by the clinical dementia rating scale that was developed here by John Morris. So thank you to John Morris and the ADRC. And then something that I'm really excited about is the registry, which is accessible through our website. We expected, I don't know, maybe 50 people or so to sign up. This number is, I think, over 1,000 now. We've been very successful recruiting people into our registry. Almost half have been diagnosed with RBD. And about 1/3 of them are women, and because our cohort is predominantly men, we are interested in trying to have more gender parity in our cohort, so we'll be recruiting more from our registry as we are able. The average age and race profile is actually very similar to our actual cohort. In terms of what we've done, we have 819 visits in the can. It's a lot. In NAPS1, we ended up seeing some people back actually annually, although that was a cross-sectional study. And in NAPS2, we're just over 200 visits. So we've been really successful collecting these standardized data, and this is the largest data set in the world on RBD that's collected in a standardized fashion. The first data freeze, which was completed by the data core led by Kenji Zhang here in statistics, the data freeze was collected, and we anticipate lots of papers will be coming out based on that data. To go back to Alzheimer's disease, which I didn't forget about, I still have a very active program in Alzheimer's disease. So one study that's ongoing is called BASE, which is Biomarkers of Alzheimer's Disease and Sleep and EEG. And the purpose of this study is to be able to use sleep and sleep-related data, hopefully collected at home, to find who has preclinical Alzheimer's disease before they have symptoms. What we do is we have people wear an actigraph at home, we do a home sleep test, we do various cognitive tests, and then they come in overnight to do an in-lab sleep study. We also collect their saliva, lots of blood, and then we collect their spinal fluid for their Alzheimer's disease biomarkers. We should be completing the study in the next 6 months or so, and I hope that we have very promising potential EEG biomarkers that we can extract from the sleep studies, both in the lab and at home, which we hope we can develop into screening tools that are non-invasive, no needles. Also, those participants from the night ADRC that we saw 10 or more years ago, these are 10 to 12 years ago, we're bringing them all back. There were 205 of them to start with. We have brought all but 4 of them back. They're being scheduled. And again, we're having them wear an actigraph, but we're also getting a sleep study this time. And I think that this study, because of the duration of the study, as well as because we have biomarker information back then and now, will be able to tease apart which direction the Alzheimer's disease pathology and the sleep relationship is going. The other sort of active activity that I do is co-leading COBRAs with Eric Music. COBRAs is a Center on Biological Rhythms and Sleep. I made up that acronym, and I feel like that is like the apex of my career. Well, now the professorship. So COBRAs, the mission of COBRAs is to bring together sleep and circadian researchers from across campus. We have over 100 members from all of these different departments and disciplines. We also have partnerships with the ADRC, the ICTS, and the Hope Center. While we like everyone in COBRAs, I think the most interesting to me is the work that we're doing in neurodegeneration and sleep. And Eric and I recently submitted a large grant to support COBRAs, as well as some very interesting projects on sleep and Alzheimer's disease, including a clinical trial that will be led by Brendan Lucy. So that's what we're doing now. In the next 1 to 11 years, I put 11 because that's when I'll be old enough that WashU will allow me to retire. I hope that we can answer the sleep Alzheimer's disease chicken or egg question as to which came first. Also, we hope that we can use sleep, circadian, and brainwave markers, ideally collected at home, to detect preclinical neurodegenerative disease, not just Alzheimer's disease, but some of these other neurodegenerative disease, and hopefully fast track them into treatment. And of course, if we're going to treat people, we need something to treat them with. And so I hope that in the next few years, we'll see clinical trials targeting sleep to reduce risk of Alzheimer's disease, and also clinical trials for RBD, for prodromal synucleinopathies, to prevent these synucleinopathies. So with that, I will end the science part of my talk, but I do have several thank you slides to go through. This is my team. They're all sitting up there. This is not a shampoo commercial. Everyone just has really good hair in my lab for some reason. I will point out that we do sleep studies overnight when most people sleep. So a lot of the people in my lab work overnight or really early or really late. So they are incredibly dedicated and hardworking, and none of this work would be possible without my lab. So thank you so very much. Also, I wanted to thank my colleagues in the sleep section who have allowed me to cut back on my clinical patients so I can devote more time to research. And this is us when we're awake. I wanted to thank the people in the NAPS consortium, particularly Brad Bove and Ron Pastuma, who are my co-PIs at, why is it verifying Microsoft Outlook? I don't want it to do that. And Ron Pastuma at McGill. This lists just the investigators involved in the many sites and cores. However, there are also many coordinators and other staff involved in the NAPS consortium that I want to acknowledge. Why is this? Again, I wanted to thank the Morris Burton family for endowing this professorship. I want to thank my research participants, many of whom are my patients or patients of my colleagues, for devoting their time to research. I wanted to thank David Holtzman, who was my Key Award mentor and continues to be really great example of a physician scientist. John Zempel was also my co-mentor for MyK, Eric Music and Brendan Lucy for their collaborations. I wanted to thank all my WashU Neurology colleagues, mentors, and letter writers. I realize I've been here a long time. Many people have written me many letters to be promoted at various levels, especially Ann Cross, who wrote a letter for me to join faculty, and at the time, and for a long time, was the only woman who was a full professor in neurology. So thank you to Ann Cross for being an awesome example. I wanted to thank my colleagues in COBRAs, and then, of course, all of the funding agencies, especially the NIH. If you're a US taxpayer, thank you. You have helped fund this research as well. Lastly, I wanted to thank my friends and family who are either here or joining online. And most of all, I wanted to thank my children for bringing joy and love and energy into my life. And then I wanted to end with a joke, which was made up by my son, and this could, I think, only be a joke made up by a child of 2 neurologists. So let me figure.
When your head is heaving, what's the worst medicine to take? Poison ibuprofen.
When you have a headache, what's the worst medicine you can take? Poison ibuprofen. All right, thank you very much.
No, I just want to congratulate you, and I think rather than having any questions, we have a reception outside, and let's go out there. I'm just afraid that if anybody here were to ask you a question, it would be, when do you sleep?
I prioritize sleep. It's good for your brain.
It's fantastic what you've done here.
Thank you.
And it's so much the Department of Neurology, the best neurology department in the country, and it's moving in a direction that is likely to help many of us. So thanks again for your great.
Oh, right, okay. Thank you, everyone.